Okay. So one of my favorite things about today's speaker is how direct we are with each other, and I was instructed quite directly to keep this brief. So don't let my brevity reflect the extent that Dr. Tseng has influenced the field and myself. So it's my absolute pleasure to introduce Dr. Gea-Ny Tseng who's a professor of physiology at VCU, not too far away. She began her journey in Taiwan, earning a Bachelor of Science in Pharmaceutical Studies and Pharmacology, and then actually moved to Colombia, where she trained with Dr. Witt. And her pioneering work, honestly, in the field of electrophysiology has been quite impressive since. She went on to move to VCU, where she was full professor since 2005. And really internationally, we know her for her work on cardiac ion channel physiology. And this consistently funded career from AHA to NIH covers structural biology, trafficking, modulation of currents, the whole deal. Her approaches are very state of the art and up to date. She incorporates electrophysiology, trafficking, biochemistry. You're going to see some amazing imaging, I'm sure. What I like about Gea-Ny is that she's literally elbows deep in the work. She's an extremely talented experimentalist who's still actually contributing to the data generated from her lab. Am I going this way now? So apart from her funding, she's also given back in terms of service, working on many AHA and NIH panels. Specifically, we're going to hear about how ion channels get where they need to go and how they work, not just the channel itself, but also the scaffolding proteins that take them there. I think that, another mention I would like to make is her commitment to mentoring and training. She also directs an undergraduate program like we have here right now, directed by myself, which is great to share that passion for mentoring. And many of her trainees have gone on to extensive careers in both clinical and research fields. So finally, just to say, apart from research and to go back to that mentoring, Gea-Ny's made a major impact on myself as well. And I think that apart from direct scientific conversation, there's also sage advice and direct advice in your career. And so I think for trainees, you don't stop needing those mentors as you become a professor. And Gea-Ny's been wonderful for that for me myself. So I'll just hand it over to her now. I look forward to your talk.
Thank you. Thank you. Can you hear me well? I would like to begin by thanking Dr. Fralin for the invitation and for Jamie for this really kind introduction. It is a great honor for me to come here and present work in my lab to this prestigious group. So this is the title of my presentation. I want to talk about how myocytes regulate channel distribution and density on the surface. I want to begin by showing a few images to give you a flavor of what I will tell you today. Here are one myocyte stand for total NAV 1.5 and surface NAV 1.5. This is a quiescent myocyte. And here is another myocyte after pacing for 15 minutes. Again, total NAV 1.5 and surface NAV 1.5. I want to, my hypothesis is that there are huge intracellular reservoirs of ion channels. That is a warehouse of channels. The newly translated channel will go there and stay there when they are needed. They can be deployed to the cell membrane from this reservoir to surface. And I also want to hopefully convince you that when we talk about the regulation of channel distribution and density on the myocyte surface, is probably we should pay more attention to the trafficking from the intracellular reservoir to the surface instead of the total channel in the cell. So before I begin, I would like to acknowledge my collaborators, Ming-Jan, Titus, VCU, Isabel introduced me to the field of sodium channel, and then Trixie, proteomics, this is also an important area my lab is going. Okay, talking about my making ion channels. It involves a lot of steps, starting with very small ribosomal RNA activation, messenger RNA bonding, assembly of the large ribosomes, translation at the rate in mammalian cell. Oh, sorry, keep on giving all my secrets. And then ER docking for channels, folding, assembly, trafficking for many of the channels, but not all of them. They need to go to Golgi for processing and then package it again to the surface. So when we talk about making of ion channels, the process can take tens of minutes or even longer. So it is good for long-term adaptation for chronic situation, but it's not fast enough for acute response. A case in point is response to a beta adrenergic stimulation. And since I'm going to focus on two channels, NAV 1.5 and slow delay rectified channel, I want to show some data, published data, illustrating how important it is for the channel to traffic in response to beta stimulation. So this is a graph from Dr. Gerdy's lab a few years ago, just to make the point that sodium channel needs to be enriched in the intercalated disc region where they are important for impulse propagation in the heart. Sodium channel respond to beta stimulation in many different ways, gating, peak phosphorylation, and whatnot. But one of them is increase the current on the cell surface. So this is a pretty early work on rabbit ventricular myocytes recording native NAV 1.5 from the cell. Application of isopaterenol to stimulate the beta adrenergic receptor cause an increase. This is inward current, so it's increase the current, reaching a plateau in 5 minutes. So this fast response likely will involve trafficking of ready-made channel from the reservoir to the surface. Another channel, the slow delay rectified channel, is a combination of KCNQ1, a voltage-gated potassium channel. It's very small in amplitude. We never see this current alone in any cells. It needs to be associated with small proteins, for example, KCE1 in the heart, to generate this very large, nice, slow delay rectified current, important for action potential repolarization. So these are traces of action potentials and then used to induce this current from guinea pig ventricular myocytes published by Tseng lab. Under basal conditions, this very long action potential in guinea pig ventricular myocytes, repolarizing is mainly induced by IK1 and IKR. IKS is very tiny, very small. But upon beta adrenergic stimulation, IK1, IKR stay about the same, but there is an explosive increase in IKS, and that is responsible for action potential shortening during beta stimulation, which is very important when we are doing exercise. We need to have shorter ventricular action potential to keep the tremble feeling. So there are several mechanisms responsible for this explosive increase in IKS during beta stimulation. One of them is trafficking of IK channel along microtubules, illustrated by this paper published many years ago. Again, it's native IKS in guinea pig ventricular myocytes, in just normal myocytes, using CK-AMP analog to activate PKA. There is a very huge increase in IKS. However, if the myocyte has been pretreated with colchicine to depolymerize microtubules, the response to CK-AMP or PK activation is very much blunted, suggesting a trafficking mechanism involved in this IKS increase. So during this presentation, I want to deal with 6 questions. I will not read them now because they will show in the slides leading to data to address these questions. But before I do that, I want to say a few words about the methods. We do most of our experiments in adult ventricular myocytes, isolated from rat or guinea pigs. We like to focus on native channels in freshly isolated myocytes as much as possible. But in cases, we want to also use adenovirus to express channels with fluorescent protein or epitope tagged, so we can manipulate and track them. And we use COR7 cells or HEC293 cells for proof of principle experiments. Experimental methods, we do a lot of imaging and data quantification. That is mainly what we do. And so I want to use this slide to explain how we do our experiments and how we analyze data. So I use NAV 1.5 in mouse as an example. We can use a so-called SCN5A external antibody targeting an extracellular epitope exposed to the actual surface of sodium channel. We can use another NAV 1.5 internal antibody generated in guinea pig to label the cell after permeabilization. So how we do this? We fix myocytes but not permeabilize the membrane, and then we incubate with first SCN5A antibody and second antibody label surface channel. And then we apply the guinea pig antibody in saponin buffer to permeabilize the cell membrane. And then, oh, sorry, what did I do? Okay, then we use this Alexa 560 antibody to label anti-guinea pig to label the total NAV 1.5. And this is what we will see with surface labeling of surface NAV 1.5 and total NAV 1.5. And with this data, we then do our Z-stack imaging and Z-stack projection. And then we are thresholding the signal, count the particles, because cell surface channel usually is in puncta. It's not homogeneous. So we can calculate the percentage of cellular area occupied this surface channel puncta and normalized by the total channel expression to give us the degree of surface expression on the different conditions. Am I going too fast? Okay, thank you. So question number one, how to differentiate between surface and intracellular channel? Again, going to Q1 GIP. The GIP tell us the total protein. And an antibody, again from Alamo, targeting the extracellular loop of between S1 and S2, allow us to detect surface KCNQ1. For the completeness, because I really need to bring this protein KCNE1, we also have this red fused to the C terminus of KCNE1 and an HA epitope inserted into the extracellular region, so we can use HA antibody to detect surface KCNE1. So we have validated these constructs in red myocytes that does not express IKS without transduction. Expression Q1 GIP and HAE1 this red give us very nice IKS current, similar to the native IKS recorded from guinea pig ventral myocytes shown here. In addition, the distinctly different distribution pattern of E1 and Q1 are also similar to those of native E1 and Q1. So let's then talk about validating this antibody. Use COR7 transfected with Q1 GIP. The fluorescence protein tells us that cell 1 has a lot of Q1 GIP, but cell 2 not. Incubation with this Q1 I'm giving all the secret again. The Q1 external antibody, we have this signal from cell 1, but not cell 2. Now, in unpermeabilized myocytes, Q1 external antibody gives us very patchy distribution, not even at all. Very different from the GIP fluorescent protein, which is the total protein. And then, but if I permeabilize the cell membrane and apply the antibody again, now the striation, the enrichment around nuclear envelope are similar to the total protein. Therefore, comparing using this case, surface versus total KCNQ1 reveals a huge intracellular reservoir of this channel protein. But that is not a unique finding with adenovirus-mediated expression of Q1 GIP, because native Q1 has the same behavior. So these are data from canine ventricular myocytes or ventricular myocardium expressing native IKS, very large, slow current. Native Q1 has this very strong striation pattern. Native E1 label mainly the cell lateral surface. And this is similar with ventricular myocardium without cell isolation, meaning that this is not a cell isolation artifact. This native Q1 in striations, not accompanied by a KCNE1, represent Q1 in intracellular reservoir. About NAV 1.5, I think I showed you some of data already, but let me go back to validation. To validate this NAV SCM5A external antibody, we created a construct of NAV 1.5 with M cherry fused to the N-terminus, flag epitope inserted into the loop or extracellular loop in domain 1, and we have validated flag antibodies from different hosts for detection. So, and this M cherry flag antibody, sodium NAV 1.5 generate inward current the same as sodium NAV 1.4 wild type. And in myocyte expression, the M cherry fluorescent protein showing the striation, the ICD concentration, and occasionally lateral surface, very similar to the native NAV 1.5 labeled with this allomon antibody targeting intracellular domain. So this construct is an authentic NAV 1.5, not being bothered by the flag or the M cherry. With that, then we use ventricular myocyte myocytes expressing this M cherry flag NAV 1.5. The external antibody applied to unpopularized myocytes give us this patchy distribution, very similar to the flag mouse antibody applied to the same cell before polymerization. And then the M cherry fluorescent protein, we don't need to polymerize the cell, shows the total protein, very different from the surface protein. And then we use mouse myocytes for freshly isolated in KB medium, detecting surface NAV 1.5, total NAV 1.5, and then the difference between the two tells us that there is a large intracellular reservoir of NAV 1.5 because in this striation pattern. And then question number 2, where are these intracellular reservoirs in this striation pattern? I think some of you may already know, but I'm going to show the data anyway. So it should be a membrane environment, large volume in the cell, in close contact with the sarcolemma for rapid delivery. And you guess it, it's the ER occupying the whole cytoplasm volume, in this case, sheep ventricular myocytes, and with junctional SR very close to sarcolemma, forming frequent contacts. But can we experimentally test that intracellular reservoir in the striation pattern is really junctional SR, but not T tubules? Let me show you the data. Q1, yeah, T tubules or junctional SR. This is something we have argued back and forth with our reviewers, but let me show the data, see whether I can convince you. So let's begin. There are 3 sets of experiments. Let's begin with the easiest one. For example, in atrial myocytes, there is no T tubule, at least in this case, shown by this membrane marker WGA, no striation. Native KCNQ1 has very prominent striation. So you don't have to have T tubule to have striation in the channel distribution. On the other hand, the striation of Q1 is very similar to that of our R2, calsequestrin 2, junctofilin 2, SERCA2A connexin, those SR or junction SR proteins. The second set of experiment is to express these Q1 GIP and E1 dysreg in canine ventral myocytes, and then immunoprecipitate them and see what proteins are present in the immunoprecipitates to deduce the environment the channel was in the cell. So let me just focus your attention to the lower panel and the IP lamp. In GIP nanobody IP Q1 GIP, we found RYR2, SERCA2A, calcium question 2, calnexin, and jongliflin 2 abundantly present in the immunoprecipitate, but they are very weak or not, almost not detectable in E1 dysreg immunoprecipitation, suggesting that Q1 GIP, when in myocytes, are probably in an environment close to or in the junction of SR. But I want to show the third set of experiment, which I'm really proud of. Here, we want to, separate sarcolemma and sarcoplasmic reticulum vesicles and use immunoblot to check whether Q1 is more abundant in one and E1 is abundant in the other. So this is my friend Minzhen. We spent a whole summer 2016 to do this experiment, and try to optimize the condition. But so we isolate hearts from guinea pigs, homogenize the hearts to generate a mixture of vesicles, some from sarcolemma, and sorry, I'm giving all my secrets again. I'm going to hide them. Okay. Sorry. Yeah. So then we incubate the vesicles with calcium oxalate with ATP energy source for calcium pump at 6 degrees Celsius. We took us a whole summer to work out. We only need 5 to 10 minutes incubation to create differential distribution. In SR, with higher calcium pump activity, they will pick up more calcium oxalate. It becomes heavier. Sarcolemma vesicle will have less calcium pump activity and pick up less calcium oxalate. So we then run sucrose gradient, and the dirty finger, it's mine. I was so surprised, so happy to see the data. I said, no, I'm just going to take a picture before we move on to the next step. So we isolate the interface A, B, C, D from light to heavy fraction. So localize the fraction and then run Western blot. And here is the results from higher, lighter fraction to lower, heavier fraction. Ranolidine, SERCA2A, calnexin, more abundant in the heavier fraction, indicating enrichment in the SR vesicles. Sarcolemma marker, the glycosylated NAC, sodium calcium exchanger 1, sodium potassium pump, more abundant in the lighter fraction than heavier fraction. Q1, more abundant in the heavier fraction. E1, more abundant in the lighter fraction. And this is the data summary. So we conclude that the striation pattern of native Q1 in myocytes is not T-tubule, it's mainly junction SR. How about NAV 1.5? I haven't done the SRSL vesicle yet. So let me just show you some simple data. But I think it's similar. In atrial myocytes, no T-tubules, as shown by this lack of T-tubule striation in WGA staining, NAV 1.5 in very prominent striation. Myocytes culture for 40 hours after transduction with GIP-packed NAV 1.5. WGA shows very little striation T-tubules, but the channel is in very abundant striation. And NAV 1.5 striation overlap with that of RYR2. And then I think this probably is a common finding for people studying NAV 1.5. We always find the channel in the ICD, lateral surface, but not always in the striation, suggesting that the striation is probably more labile compartment where channel can move in and out. Question number 3. How do ion channels reach or enter the junction SR? Well, this is a graph from my lab where we use fluorescent in situ hybridization to find GIP messenger RNA and compare that with the GIP NAV1.5 protein. You see at this stage, the messenger RNA particles and the channel are focused around nuclei. And we look at this GIP NAV1.5 expression over time, 12 hours, 18, 20, and 30, and 50 hours. You see that it begins in the nuclear envelope region, spreading out in striations toward the lateral surface and ICD, suggesting that this channel can be translated in nuclear envelope. And then because nuclear envelope is connected to with the SR-ER junction, SR is one big organelle. They don't need to enter any vesicles. They can move along the membrane in the striation pattern to other areas of the cell. And KCNQ1 likely follow the same trend, the path. So this is a myocyte transduced with Q1 GIP at different time, beginning at a nuclear envelope, moving outward in striation pattern toward the lateral surface. No vesicles, at least not prominently. However, in 2021, a paper from a group at Ohio State University suggests that in mouse myocytes, NAV1.5 translation can be dispersed in a wide area of the cell. This is based on detection of NAV1.5, native NAV1.5 messenger RNA in these cells. This is likely because we have seen that two proteins, connexin and BIP, they are chaperones involved in protein folding when they are being translated in the ER or rough ER. They actually are concentrated in striation pattern along the Z line, suggesting junction SR may have the ability to translate proteins. And I will skip this part for now. The third possibility is that surface channel endocytosis may re-enter junction SR. So I show you this protocol before with one addition. So fixed but not permeabilized external antibody staining, guinea pig antibody incubation in saponin to permeabilize the cell membrane, followed by anti-guinea pig and anti-rabbit. This rabbit can only come from here because there's no more rabbit antibody anymore. So what you are seeing here is with this protocol, we have the total protein, we have the surface protein, we have what I call endocytosis protein, matching the surface well, but also showing some striation. This is a kind of difficult because I did not allow any endocytosis. I fixed the myocytes without any endocytosis. But I think maybe even in fixed myocytes, the endocytosis can still happen to some degree. So that's it. That's the explanation I can come up with at this moment. But if I incubate the live myocyte with this external antibody at 36 degrees Celsius for 60 minutes, and then wash and then fix but not permeabilize antibody on surface, guinea pig antibody, saponin buffer, and the other two antibodies, this is what you see. Oh, sorry. Yes. Total protein. Yes.
Is there any functional difference now between the re-endocytosed NAV1.5s and those that are coming up fresh, or is there a way you can get at that?
I think the question is, can I label the endocytosis channel and then label them differently when they re-expose to the cell surface?
That would be the starting point, and I was just curious about functionality, but
right. Functionality, yes. I try to record using cell attach patch close to where the patches are using immunogold labeled with antibody has not worked yet, at least in my hand. It's just I need to troubleshoot and optimize. So I cannot answer the question of surface sodium channel recording. In terms of labeling endocytosis and resurface channel, that's something I want to do. I haven't been able to figure that out yet. Yeah. Yes.
So there are some membrane dyes that primarily label the plasmalemma. And could you do that in combination and look for the internalized membrane and the antibody label channel?
So you are suggesting cellular fractionation.
Yeah.
And then, yeah, need to think about that. I have been focusing on imaging, differential labeling, but it's certainly something we need to think about if the labeling does not work. So, So in this case, after 60 minutes incubation, actually there is very little or none surface protein, but then the endocytosis protein enter the striation pattern along the Z line, suggesting junction SR. And Q1 probably is doing the same. So here is using the no endocytosis protocol, total Q1 GIP fluorescent protein, surface Q1, very patchy distribution, endocytosis Q1, correlation, but also striation. After 60 minutes of incubation with the antibody and doing the protocol, total channel protein, surface almost all gone. I'm using the Z projection to catch some of the particles that I could detect, but endocytosis Q1 in this very strong striation. And I'm also noting that the total protein is very abundant around the nuclear envelope, but the so-called endocytosis protein is not, meaning it's just different from newly translated protein around the nuclear envelope. Question number 4, what triggers channel to exit from the junction of SR? Here, I need to bring in KC and U1 as a contrast because I think the two of them together will give me a better chance of explaining what I think happened. So if we look at this surface Q1 in this case, and surface E1 shown here, and this is the total channel, you immediately see that this is very homogeneous around the cell surface and very different from the patchy distribution of Q1. And so a tale of two channel proteins. So if we look at this, again, the virus, adenovirus transduction and time course of expression, you see that in the case of Q1 GIP, nuclear envelope, striation pattern distribution and surface, but E1, nuclear envelope, but very quickly entering vascular compartment. And then very strong, and then gradually missing or disappearing from the vascular compartment, showing in the lateral surface. So coming back to this scheme, in myocytes, you can have proteins translated junction to SR, in vesicles moving to the Golgi, and then entering the Golgi, shown by this WGA and beta cope staining. So when we compare E1 this red with WGA and beta cope, those vesicles correspond 100% with WGA signal. Whereas with Q1, nuclear envelope, striation, no vascular compartment. So we suggest that E1 follows this conventional secreted pathway, ROF ER, vesicles, Golgi, surface, but Q1, not so. And I want to show you one very cute strategy to make this more concrete. This is called retention using selected hook rush. What it is that we will create a rush plasmid with our protein of interest fused with this red component, yellow component called stratavidin binding peptide, and another ER resonant protein called ER hook fused with SCD, strata within core domain. They were expressed as two separate proteins. So when they are expressed in cells, the protein of interest will be hooked at the ER by binding of the yellow component to the red component. Adding biotin to the cell, biotin will diffuse into the cell, binding to this red component, displace the yellow component, allowing the protein of interest to depart from ER because it's not hooked anymore and moving toward down its secretory path to wherever they need to go. So I want to show you two sets of experiments. The first one is doing ER hooked HAE1, this red, just ER hooked E1 will be fine, and ER hooked VSVG GIP. VSVG is the model molecule that cell biology people have used to track constitutive trafficking from rough ER, is a membrane protein, to the plasma membrane. So we compare our E1 with VSVG to see how similar, how different they are. So before biotin, both are ER hooked in the ER in this pattern. Half an hour, 60 minutes, 2 hours, you see they are moving away from ER into the Golgi compartment in vesicles, and then getting into vesicles, moving to the cell surface, suggesting that in the same cell, E1 is trafficking just like the constitutively trafficking VSVG from ER to the cell surface. How about Q1? We have ER hooked Q1 GIP, in this case, co-expressed with DR. This red labeled ER marker is not E1, it's an ER marker. So this is before biotin, 30 minutes, 60 minutes, 120 minutes. It does not like to leave ER, it stays in the ER. So we suggest that E1 follows the conventional pathway from rough ER through Golgi to plasma membrane constitutively, to the membrane constitutively, whereas Q1 appears to follow an unconventional pathway. And there are several different flavors. Which one is the one that Q1 follows? So we set up to hunt for, my hunch is that Q1 may be leaving ER in vesicles, reaching the cell membrane directly, not going through Golgi. We actually try, there is no glycosylation with Q1, although a very strong glycosylation signal with Q1. So let me just focus on the third set of experiment. Here we have Q1 GIP co-expressed with D1, this red labeled E1. We switch between TURF imaging to look at fluorescent protein close to the bottom surface of a cell, and the wide field imaging split camera setting. There's a time lag between the two of 50 microseconds, so they are practically simultaneous. So time 0, we have vesicles close to cell surface, and ER network deep into the myocyte, in the cytoplasm. We have two types of vesicles labeled. The yellow arrow points to these vesicles that do not move. But the interesting one is the one labeled by these green arrows. Next screen, after 0.65 seconds, you see that that vesicle becomes stronger. Third scan, 1.3 seconds, is gone. It's compared with some other some other data, led us to propose that Q1 can directly exit ER to surface without going to Golgi. And this is not constitutive. It likes to stay in the ER, does not want to come out. So, but we still have Q1 on the surface. We can do patch clamp to see that. So there must be a Q for Q1 to exit the ER. What is the Q? So I think I think we will be fine. We'll be fine, right? So going back to really early, before some of you were born, I'm sure. There was this very nice paper published from Toshi's lab recording IKS. Oh, sorry. Okay, sorry. In guinea pig ventromyocytes, where he used pipette solution switch method to elevate the calcium concentration in the cytoplasm, and he saw very nice increase in IKS. Then cryo-EM structure shows that there are 4 molecules of comodulin attached to the Q1 cytoplasmic domain constitutively. And 2 papers published in 2006. I cite one expression in oocytes, Q1, E1 together, very nice IKS, injecting beta into the cell to reduce cytoplasmic calcium, reduce the current. Q1 alone, similar, beta injection, reduce the current and create this very interesting kinetic component. But the most important one is this one. Q1 co-expression with white-type comodulin or with a mutant comodulin, that cannot bind. Oh, sorry. Calcium with much more smaller current and that very interesting kinetic component. So it's calcium bonding to the comodulin that can promote Q1 to the surface. I think I will just skip the following few slides from my own lab. The conclusion there is that with in core7 expression, the angiotensin II receptor stimulation can increase surface Q1 through a calcium comodulin binding mechanism. And now, how about NAV 1.5? Going back to that 1992 paper, guinea pig, rabbit ventromyocyte, isoprotein increase the current. They use inside our patch to test how catalytic subunit of protein kinase A can influence the channel. There's an increase in single channel activity. So it's a pKa mediated event. And years later, almost 10 years later, work from Murray's lab showed that there are ER retention signals in the domain 1-2 loop of sodium channel. pK phosphorylation of nearby residues generated by the negative charge will mask a positive charge of ER retention, allowing NAV 1.5 to exit ER to the cell surface. But that is pKa phosphorylation or pKc phosphorylation. How about without phosphorylation? We know that there is comodulin bound to the C-terminus of NAV 1.5. So we did a similar experiment as I skipped a few moments ago. Co-expressing GIP NAV 1.5 AT1 receptor in core7 cells, along with wild type or the mutant comodulin. This does not bind calcium. Control or angiotensin II 1 micromolar 1 hour, and using biotin to quantify surface NAV 1.5, using EB1 to show that surface protein has very little contamination from cytoplasmic protein. And here is our calculation. Let's just focus on the second row here. Compared with control, angiotensin II increased surface NAV 1.5 by more than 2-fold. However, if I have the mutant comodulin overexpressed, preventing calcium binding, then angiotensin II treatment does not increase cell surface NAV 1.5, suggesting a similar scheme. Let's move on to, we have 2 more questions. Question 5, what regulates channel density on the myocyte surface? If we think about the location of the reservoir, the Q1 and NAV 1.5 in the reservoir in the junction SR with comodulin exposed to the didactic space between plasma membrane and junction SR membrane, then is it possible that if we paste the cell, I'm talking about myocytes now, to cause calcium-induced calcium release, could that enhance channel trafficking to the surface by calcium binding to comodulin? exposed to the space between T-tubule membrane and junctional SR. So this is the question we ask. And this, I think, is get to the, what I would like to propose is really the trafficking between the reservoir and plasma membrane. That's important for the surface density. So here is the data, just big summary of sodium channel NAV 1.5. Surface, total, no pacing, pacing 15 minutes, pacing 30 minutes. Surface NAV 1.5 increase during after pacing, but total channel pixel content does not change. In this case, I was using pKa in my medium, activation in the medium. I found that if I don't have pKa activation in these myocytes, I can stimulate with field pacing. I can pace about 40% or even fewer of the myocytes. But if I have pKa to enhance the calcium uptake and release, I can have up to 90% cell being paced. So I was using that to help the pacing protocol. So, so this is NAV 1.5. And with a pKa is similar. Let me just focus on the left part. Oh, this is the pacing for 15 minutes. If I have pKa pacing more efficient, I have more surface Q1 than pacing without pKa activation. And this is only in the presence of KCNE1, not Q1 alone. It's a big question, and I don't have the answer yet. But this is, I was expecting to, I expect to see increase of Q1 on the surface without E1, because that was the theory. But in the myocyte, for whatever reason, the behavior seems to be different from COS7 or oocyte expression. So there are things to work out with myocyte environment. So before I get to the last question, I want to check whether we need actin filaments or microtubules for channel to traffic to its reservoir, to the surface. And these are just orthogonal view image of NAV 1.5 Q1. Then the channel needs to traffic a few micrometers. It's not immediate jump across the dilated space. And here is the summary of the data, very recent data. I treat the cells with langtronculin A 5 micromolar for 2 hours before pacing with pKa, or I treat the cell with nocodazole to depolymerize microtubule for 2 hours before the pacing. Then I have, of course, I have the control. I start pacing the 3 groups, and I count surface NAV 1.5 or surface Q1 after this disruption of the actin filaments or microtubules. And you can see very clearly this reduction of surface NAV 1.5, surface Q1 GIP with disruption of the actin filaments and microtubule. And I want to focus on the pacing because I want to see whether this cytoskeleton trafficking is needed for channel moving junction artery membrane. So getting to, oh, sorry about that. So number 6 is something we need to work out, and we are not there yet. We saw that the surface channel is in patches. It's not homogeneous. What determines where they go when they leave junction SR? I think, or I believe, or my hunch is that the microtubule plus and binding protein, EB1, is important. EB1 works as dimer along the cell periphery that has been shown to be involved in channel trafficking, connexin 43 and NAV 1.5. And a little bit complicated data from my own lab, we show that Q1 in the form of Q1 GIP, E1 in the form of HAE1 distress only the Q1 can interact with native EB1 in myocytes, but not the casein E1. So again, apology for this complete, 就是whole cell lysate, negative control, positive control. Q1 immunoprecipitate itself, immunoprecipitate E1, and immunoprecipitate EB1 dimer. HAE1 immunoprecipitate Q1, E1, but very little or none BB1. EB1 precipitate Q1, but not this red E1 and dimer. So there's a differentiation in EB1 interaction. And here is the final data slide. With surface NAV 1.5 in the form of M-trait reflect NAV 1.5, surface distribution have some correlation to subsarcolemma EB1. Likewise, Q1 GIP, surface patches has some correlation to the EB1 beneath the cell membrane. Whereas HAE1, this red, that will consecutively go to the cell surface with almost homogeneous distribution. There is no correlation with EB1. We are in the process of immunoprecipitate EB1 and use proteomics to find EB1 interacting proteins in myocytes. Not easy thing to do. And so I don't have data to show you yet. This is just taken from a website to show that in left ventricles, according to the literature, there are dozens of interacting proteins that can interact with EB1, or also called MAPRE1. So we have a lot of targets to tease out and work with. So with that, I want to summarize what I have told you. Let me just read the text. Q1 and NAV1 have patch distribution on the surface, very different from the total protein. And since we have all been studying total protein distribution, I think we want to pay attention to the surface pool. And Q1 and NAV1 have a large insulin reservoir in the junction SR. Newly translated protein can be stored in junction SR before deploying to the cell surface. Calcium elevation during EC coupling may trigger Q1 and NAV1.5 junction, exiting junction SR, trafficking on filaments, macroactin filament on the tubule to surface. And those cytosine Q1 and NAV may enter junction SR reservoir. And EB1 may guide this protein from junction SR to myocyte surface, although I'm sure many other proteins are involved in this guide, the trafficking process. And we are also interested in knowing whether other voltage-gated channels, calcium channels and hERG channels, both have chemodulin associated with their cytoplasmic domain, may also traffic in the same fashion with this intracellular reservoir and traffic to the cell surface upon calcium elevation during EC coupling. So thank you for your attention.
Questions? Alex? So that was all really fascinating work. Thank you so much for sharing that. I know a lot of, all the work was done in isolated ventricular myocytes as well. Have you looked at like NRVM, like neonatal rat ventricular myocytes, or any other cultured cells that have connections to see if they still have this sodium channel reserve? I'm wondering if it could be something that these cells are already expressing, a large amount of the surface when connected or forming intercalated discs with one another, and pacing the cells might be sort of rescuing the phenotype.
Very good question, thank you. Yeah, I think you are getting into the key point here. I'm only using isolated myocytes. Am I still seeing artifacts in the whole heart? Do we have more homogeneous distribution on the surface? Your idea is actually a very good one, doing neonatal myocyte in cell monolayer, where I can have access to the surface to detect. Because I have been thinking about how we can do this in myocardium without cell myocyte isolation. I cannot cross the barrier of permeabilization necessary for antibody access. But I think neonatal myocyte will be something we are going to try. Thank you.
Mark?
The calcium elevation regarding the calcination that stimulates the traffic to the membrane. I was wondering if that's rate dependent, and that would be interesting for arrhythmia, right? It is also mediated by CAM kinase, which is like a rate sensor. That's one question. And the other one is regarding, it's maybe kind of a bit similar to what Alex was asking, is regarding the isolated cell model. And you said it was you required to have PKA activated, and in for pacing to induce this trafficking, right? And in the whole heart, we know that PKA is constitutively active. So I was wondering if that's a technical thing, or what is the reason that you have to activate PKA in the isolated cells?
Oh, let me answer the easier question. For the pacing experiment, I just compare where I'm using the Alnox optics, A-well format. If I don't have PKA activator, CK-AMP, permeability CK-AMP, alkali acid, just control medium, I can see only 30–40% cells contracting when I pace them. But if I have PKA activation, I see 80–90% cell activating. So I need to use PKA activation, and that's probably is enhancing the calcium uptake and release, allowing the cell to contract. Without more data, I don't know the mechanism, but it's just higher percentage of cell respond to the pacing and start contracting, making the data immunostaining and quantification easier to interpret. But if you tell me that there is a basal level PK activation in a beating living heart, then it makes sense. Then I'm probably not guilty of using PKA to enhance.
We block PKA, we see a huge change in conduction. So that's kind of the idea. That's why I think it's active. It's constantly active in the whole heart. So
now about pacing frequency, I was using 0.5 hertz pacing in all my experiments. I have not tested pacing at different rates. In my hands, when I pace mouse for longer time, I lost mouse. They just drop out. Probably the electrolyte imbalance and whatnot. So half an hour seems to be the optimum time. I see a lot of cells beating and not much drop out. But I extend to 60 minutes, 120 minutes, and they were not happy then. So, but it's not the frequency. I think it's also important and useful to test different frequencies, 1 hertz or even faster or lower, to see whether I can gauge or just grade it, enhance our surface. Yes.
I'll try and pack in another couple of questions here if we can. So Anthony, yep, cool. Yeah, that's. Oh, sorry.
Okay. So I'm wondering about the relationship of newly synthesized, newly translated channels to ones that are currently available, because a lot of the experiments you're doing, you assume they've already been, transcripts have been translated in your. So is there a scaffolding mechanism to move message selectively to the junctional ER? And then does that actually also influence the membrane trafficking of the?
It's a question about why they want to go into junctional SR. Yeah, I think this is my thought, but I may be very wrong, and I happen to know all the critiques. Proteins the channel proteins are translated at rough ER. That can be nuclear envelope, but there can also be junctional SR because the machinery seems to be there. So they are locally synthesized or moved to junctional SR. Junctional SR is a good place to be because it's so close to the cell membrane. So you don't really need to travel very long distance, for example, from the deep ER or SR to the membrane.
RNP or something like that.
RNA
binding.
Postcodes are great. RNA binding protein to take it.
Yeah. Oh, okay. Yeah. We did some fish experiment, and Ohio Group also did fish experiment. And I don't think we have, or they have looked deep into the trafficking mechanism for RNA. Yeah.
Okay, Mike.
Yeah, I was wondering about your very first or second slide talked about with the beta adrenergic stimulation, the explosive change in the current. So when I'm trying to put this all in context, and I'm thinking about, for example, single channel conductances due to phosphorylation events and changes, and now you're kind of swamping the surface with all this new pool of channels. Is there, I'm trying to think about how to put it together over time. Is there any way for specificity or targeting in terms of the replacement of the membrane for channels in one state versus another? That is, those that have different phosphorylation events have changed their single channel conductance, or is it just a willy-nilly replacement or addition to all the channels?
Let me, if I may, rephrase the questions to see if I understand. So there are surface E1 kind of homogeneously on the surface, and there are patches of Q1 on the surface. When we have beta stimulation, I'm proposing that there'll be more Q1 moving to the surface, joining with E1 to form more IKS. Is that?
Yeah, more or less. But in terms of individual channels or groups of channels that have changed their conductance state, I don't know if they're clustered together or if they're evenly distributed to begin with. I mean, maybe that's the heart of the question that I'm trying to ask you. How does that happen?
I think I think the single channel conductance will remain the same. The clustering is just they are where they are, and they may influence each other by so-called positive, something about positive. This UC Davis group suggests that when channels form clusters, they help each other to open longer. But the single channel conductance, I don't think will be so easily manipulated.
Yeah, I just don't know if the phosphorylation events for that are occurring locally in those clusters, and if there's some crosstalk, their cooperativity that you're alluding to.
Yes.
Anyway, okay, thanks.
Sorry. I hope
real quick, Rowan, real quick. Sorry, Greg.
So excitation contraction coupling is cyclical. And I'm having a little bit of trouble understanding why, with the calcium elevation, it triggers the trafficking to the membrane. Is it like an ebb and flow with excitation contraction coupling? Why would it go to the plasma membrane if it's not also coming back from the plasma membrane and recycling?
I think what I have in mind is that in a beating normal heart, there is a cyclic calcium-induced calcium release in that dielectric junction. And that, I think, may provide the basal force mechanism for channel to leave junction SR to the cell membrane, but not too much, just enough to sustain the channel function and maybe 1.5 or Q1 or IKS. But if you have beta stimulation, where you have more calcium release during EC coupling, then there will be more calcium bonding to the calmodulin, and then more surface expression and more currents. So I think this, I think I need to emphasize that this basal level of surface expression that may depend on calcium calmodulin, but that mechanism will, I think, really work during beta stimulation when you have more calcium release during action potential. Am I answering your question?
All right. Speaking of which, let's call it there. And thanks, Gea-Ny, again for a really